Okay, hello everyone. Welcome back. So we are moving on to week 6. And this week we're going to be discussing the cell cycle, including DNA synthesis and mitosis. And then we're also going to discuss meiosis. So today I'm only going to discuss mitosis, the first part of this lecture, or this lab, and then we'll discuss meiosis later this week. So let me go ahead and share my screen real quick. Okay. Okay, so the purpose of this lab is to learn about the two types of nuclear division that occurs in eukaryotic cells. So let me just make sure I'm recording before I get too far ahead. Okay, yes, I am. Okay, so like I was saying, the purpose is to learn about the two types of nuclear division that can occur in eukaryotic cells, and these two types of cellular division would be mitosis and meiosis. So we will not be discussing how prokaryotic cells undergo cell division. We are only going to focus on eukaryotic cells. And today I'm only going to cover the first part of this lab, which is mitosis. So I believe this includes activity 1, 2, and 3. So first I need to introduce the cell cycle. And that is what is shown here. This here is the cell cycle, and it is a series of events that takes place in a cell, leading to its division into two daughter cells. So as you can see, the major phases of the cell can be divided into smaller phases. So the major phases would be the interphase, which is shown here in this light blue arrow. And then the other major phase is called the mitotic phase, which is going to be this portion here, the M phase. But the interphase can be split up into these three phases. We have the G1, or the first gap phase, the S phase, which is our synthesis phase, and our G2, our second gap phase. And as you can see here, you start off with one parent cell. This is your DNA or genetic information shown here. And then as you progress to the S phase, this DNA, this chromosome is going to be replicated. So you're going to have a sister. So here you have one, let me start over. Here you have one chromosome that contains your DNA. This is going to be referred to as your parent cell. When you go to the S phase, you'll see that this one chromosome, the DNA is going to be replicated. So you're going to have the exact same information over here. And now you're going to have this sister chromatid. This is still one chromosome because you have one centromere here connecting them. Basically, the information here is identical to the information here. And then I'm gonna go over these phases a little bit more detail, but essentially in the M phase, you can see that this is basically going to get split into two. So as you can see here in the M phase, it's going to get split into two identical cells. So these daughter cells are going to be identical to this parent cell. So it's very important to remember that these are going to be identical. So your lab manual mentions this, so please make sure to read your guys' manual always, but it says that in actively dividing cells, this process can take approximately 24 hours to complete. So this is a pretty quick process if you think of it, and depending on what cell you're looking at, this process occurs more frequently than in other cells. So what I mean by that is that, as you can imagine, cells like the epithelial cells found on your skin, cells that line your GI tract and your urinary tract, you I guess shed those cells constantly, so those cells need to be regenerated constantly. So they're going to undergo this process a lot more. And those type of cells are referred to as labile cells. And then we have what are called stable cells. Stable cell, a great example of this would be your liver cells or hepatocytes. So your liver cells or hepatocytes, they're usually in this other phase that's over here called a G0 phase. It is a quiescent phase where essentially your cells are just relaxing and chilling and waiting to get some kind of stimulus to say that they need to go into this cell cycle or replicate themselves. And for example, if you were to donate a portion of your liver, like say you donate 40% of your liver to be used in a liver transplant, your hepatocytes would be activated. This is like an external stimulus saying, okay, now we need to replicate and get back to our normal size. And they would jump into this cell cycle, replicate until they get back to their normal size. And then they once again, so they regenerate themselves. Your liver gets regenerated to its normal size, and then it goes back to that G0 or quiescent phase, where it's just relaxing again. So back to the cell cycle. So you can essentially divide this into two phases. Like I said, you have your interphase. Your interphase consists of the G1, the S phase, and the G2. And then you have your M phase, which is your mitotic phase that we'll go into more detail later. But your M phase consists of the prophase, metaphase, anaphase, telophase, and cytokinesis. Okay. So I already mentioned this a little bit, but your interphase will consist of these three phases. Your G1 phase is your first gap phase, and this is really your first growth phase. So we're basically going from one cell to two cells, right? So let's think of what a cell is composed of. A cell is composed of a plasma membrane, it's composed of organelles, it's composed of a nucleus that contains DNA, cytoplasm. All those things are components of a cell. So in this first growth phase, basically this cell is getting ready to replicate itself. So it has to make more organelles first off, like it has to essentially double the amount of mitochondria it has, so that each cell here has the exact same mitochondria, the exact same Golgi apparatus, the exact same ribosomes, etc. So the first thing that's going to happen in this first growth phase is that you're essentially going to make more organelles. You're also going to start synthesizing proteins, enzymes, transcription factors, etc. that are going to be needed in this S phase over here. So once that's all done, you progress to the S phase. So in the S phase, here you have DNA replication occur. So this is going to be DNA synthesis and chromosome duplication. So here you also need the generation of DNA polymerase 1 and 3. They're going to be very important in DNA replication. There's also a bunch of other enzymes that are important in proteins. You have helicase, primase, all sorts of things that are required for DNA replication. And we're going to actually go into DNA replication in more detail later on. And then you have our second gap phase or a second growth phase. So here, as you can imagine, you already have double amount of organelles, you have double amount of DNA, but now your cell actually has to grow. It has to be large enough to bud off into two cells. And so it has to just generally grow, so increase its cell size and increase the amount of cytoplasm. So the cytoplasm here is even to the cytoplasm here, and it's identical to the amount of cytoplasm that it was found here. So that's essentially what's going to occur in each of these phases during interphase. So if you're starting to think like a scientist, you're going to be asking yourself, how do these cells know whether it's okay to go on to the next phase? And there's various checkpoints. So you have, I think this image might show it a little better. Oh, and this image also shows that G0, that quiescent phase that I told you that those hepatocytes just sit at and relax, do their thing until they're called into the cell cycle. Okay, so like I was saying, how do these cells know whether to go on and replicate themselves? Well, obviously they're going to get some kind of external stimulus saying, okay, we need you to replicate yourself, and it jumps into this G1 phase then. Okay, but how does it know to go from the G1 phase to the S phase? And essentially there are so much goes into it, way more than I'm going to discuss here. And as you can see in this image, it's pretty busy. I'm not going to go through all this, but it's even more complicated than it's just shown in this image. But essentially, you have genes that are going to want to stimulate replication. They're going to be telling your cell, go, 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 replicate yourself. We need more of you. And if you think of yourself like, oh, you fell off of your skateboard and some of your skin cells scraped off, like, well, I guess other pathways would also be activated in that sense. But if you flake off some of your skin, I guess, and it's not a deep abrasion, this cell cycle is going to be going. It's going to be activated. So you have genes that want to stimulate this replication, and these are called proto-oncogenes. And you guys have probably heard of these. So they essentially are going to get their input from various growth factors, various mitogens, and they're going to activate cell signaling cascades. We'll just leave it at that for now. They activate different signaling cascades, and these signaling cascades are going to make what is called CDK, which is cyclin-dependent kinase. It's an enzyme kinase. It's going to phosphorylate something, and then cyclin. So when these two are not interacting, they are inactive. When they are inactive, your cell is not going through the cell cycle. When these two interact, this kinase interacts with cyclin. It is told to go progress through the cell cycle. So when specifically CDK4 and 6 interact with cyclin D in the G1 phase, and then this CDK interacts with this cyclin in the G1 phase, it tells it to go on to the S phase. So this is basically when they're interacting, they are active and they are telling your cell to keep going, like you're on the right path, keep replicating. So that's basically how your cells know whether it is okay to go on. But there's also, this is very important, we're replicating our DNA, so there has to be some kind of checks and balance system, I guess, in our body to make sure that this isn't just going crazy, because as you can imagine, when there's too much cellular division, that leads to tumors, which you probably know of a disease that this can result in, cancer, where this process is dysregulated. So there's different checkpoints. So you can see there's this G1S checkpoint, there's this G2M checkpoint, and there's a checkpoint over here. Actually, there's supposed to be a checkpoint. I don't know if I see it. There's supposed to be a checkpoint in your M phase. Oh, right here, this checkpoint as well. So there's various checkpoints to make sure things check out and everything's okay. So as you can imagine, as it goes through the G1 phase, it reaches this checkpoint, and at this point, this is your first growth phase. So once it reaches this checkpoint, your cell is basically asked, is your DNA healthy? If your DNA is not healthy, well, then we don't want to replicate this DNA and then end up with 2 unhealthy cells at the very end, right? So at this G1S checkpoint, the first thing it does is ask, okay, are you damaged? If you are damaged, can I repair you? So these are what are referred to as tumor suppressant genes. So you have different systems here that look at your DNA and say, 1, are you damaged? And if it is damaged, it then asks, is it repairable? If it's repairable, then it repairs itself, and then it moves on to the S phase. If it's not repairable, then what happens is your P53, which is this guy here, is going to basically tell your cell to undergo apoptosis. Apoptosis is programmed cell death. So then your cell will die because the damage was not repairable, and that cell is obviously dies, so it can't replicate itself. And there are various checkpoints at various stages, and this is very important because although your cells are great at doing this, they actually go through these processes so fast and so accurately. For example, in the S phase, where DNA replication occurs, your cells are able to do this so rapidly and so accurately, but yet there is still systems in place to come in and make sure that if there is a mismatch or something happens during this replication, something doesn't get replicated accurately, something can come in and repair the damage. Okay. So this is essentially showing that again, the programmed cell death, like, so P53 is usually interacting with this other guy called MDM2. And in this state, this, I guess, pathway is inactive. When this other there's this other protein called ATM. When ATM goes and proofreads your DNA and finds an error in the DNA, ATM will come over here, take this MDM2 off. This P53 will get activated, and the P53 will go into cell cycle arrest, where it pauses the cell cycle. It initiates repair, and if it's able to repair, it goes back to the cell cycle. If it's not able to repair, it undergoes apoptosis, which is programmed cell death. And then, as you can imagine, this leads to genetic stability. Both of these outcomes lead to genetic stability. So the cell cycle is division of one cell into daughter cells. So we're only going to discuss mitosis here, but mitosis is the division of the nucleus equally to produce two identical daughter cells. So I kind of just want to go over this with you guys very briefly. I'm sure you guys already covered this, so I'm not going to go into too much detail, but in your G1 phase, you would have your chromosome. This is one chromosome shown here that is unreplicated. So you have your short arm, a centromere, and what is usually referred to as the long arm. And then once it goes into your S phase, this chromosome is going to be replicated. So you have your one here and one here. This is still referred to as one chromosome because there's one centromere, but here you have sister chromatids. This is two chromatids that are joined by a common centromere. And each carries identical genetic information. So here's a video that I want you guys to watch on your own time, just so you guys can see how the DNA is packed into these chromosomes. So the DNA, your DNA basically will wrap around these histones, these histone proteins, in order to be tightly packaged into these chromosomes. So this is like a minute or so. You could actually speed it up if you want and finish it faster. So we kind of already talked about DNA. I believe it was during week 3. But essentially here, you see that DNA is a polymer that is made up of 4 types of nucleotides. So in every nucleotide, you're going to have a phosphate group, which is shown here in the yellow. You're going to have a pentose sugar. Since this is DNA, this pentose sugar is going to be deoxyribose. And then you're going to have a nitrogenous base. So here the nitrogenous base shown is thymine, but this nitrogenous base can also be adenine, cytosine, or guanine. And as you can see up here, these nitrogenous bases come in 2 types. You can see that adenine has 2 ring structures, cytosine has 1 ring structure, thymine has 1 ring structure, and guanine has 2 ring structures. This right here where the pentose sugar and the phosphate interact is going to be referred to as the sugar phosphate background. And as you can show that sugar, as you can see in this image, that sugar phosphate background is going to be what's highlighted here in this blue ribbon. Then you have your nitrogenous bases coming out from there. So as you can see, your nitrogenous bases come out from this interaction. And they're found inside the helix. And they will interact with a complementary nitrogenous base. And the way these two are stabilized are through hydrogen bonds. So these are referred to as anti-parallel complementary strands. And I'll talk a little bit more about that. So like I said, there are different bases. You have your cytosine and thymine. You basically see one ring structure, and these are called pyrimidines. And you have your guanine and adenine, which have these purine ring structures. So let me make sure there was nothing else I want to say about this. So yeah, so your pyrimidines have a single ring structure and your purines have a double ring structure. And this just shows this scientist, Mrs. Franklin, who was the first to obtain images of DNA, and which is shown here. And these images are what led to the discovery of the double helix. So you guys probably already know this, that DNA comes in this double helix as shown in this image here. And this was proposed in 1953. And then this image, which is shown here, Franklin's DNA image, led to the discovery of the double helix model. Watson and Crick were the ones that proposed this, and they went on to win a Nobel Prize. Originally, her work was actually not even recognized or mentioned, but that has since then turned around, and she has been credited for her work in the discovery of the double helix structure. So like I said, you have your pentose sugar and your phosphate group, and those are on the, I guess, outside. We're going to call it the outside, and they make this sugar phosphate backbone, which is shown here in this pink ribbon. So your strands, they always run from 5' to 3'. So as you can see, it runs from 5' to 3'. It runs down. And then the other strand is going to run in the opposite direction. So they are anti-parallel. It's going to run from 5', which is here, to 3', which is up. So it's going to run up from 5' to 3'. And remember that the guanine will always interact with cytosine. Thymine will always interact with your adenine. And as you can see here, your adenine and thymine will have 2 hydrogen bonds between them, which is shown here. They're going to be stabilized by 2 hydrogen bonds. Guanine and cytosine are going to be stabilized by 3 hydrogen bonds. And that is going to be in the interior or the inside of this double helix. And as you can see, your guanine with cytosine, your 3 hydrogen bonds here, they're found on the inside. Your sugar phosphate backbone is this ribbon. This one runs from 5' to 3', which is down. This one runs from 5' to 3', which is up. So these are always complementary, and they're always anti-parallel. And this is discussed a little bit more here. So you have complementary strands, meaning that certain bases always interact with other bases, and these 2 anti-parallel strands are held together by hydrogen bonds. Strands are complementary. A is always across from T, and C is always across from G. So in your first activity, you're going to go ahead and I guess attempt DNA replication. So here, remember this goes from 5' to 3', 5' to 3'. You will, new nucleotide triphosphate will come always to the, and add on to the end of the 3'. And when it comes on and adds to this end of the 3', 2 phosphate groups are going to leave. So you're going to be left with a pentose sugar, your phosphate, which is going to make up that sugar phosphate backbone. Your thymine will come and interact with your A, and then 2 phosphates leave, and that essentially continues on the 3' end. Your strand will keep growing. So new nucleotides can only be added at the 3' end. So DNA polymerases are enzymes that are responsible for new DNA synthesis. So DNA polymerase can only add nucleotides to the 3' end of the growing strand. The strand is constructed continuously. And I'll talk about this in the next slide, but one strand is going to be constructed continuously, and the other strand is going to be constructed in segments, and these are referred to as Okazaki fragments, and they are joined by DNA ligase. So essentially here you have, this is all happening in that S phase. Remember, this all happens in S phase of our cell cycle where the DNA gets replicated. So our deep blue is our parental DNA, and our baby blue is going to be that the new template DNA, I guess we'll call it, or not the template, the new DNA. So our dark blue is the parental DNA. This enzyme shown here, helicase, is going to come in and basically unzip these this parent DNA so that it creates what is called a replication fork. It's going to open up and create this, I guess we could call it a bubble or replication fork. And essentially, this parent DNA strand is running from 5' to 3', and then the new one's going to be from 5' over here to 3' over here. And this over here is going to be referred to as your leading strand. Down here, this one's going to run in the opposite direction. It's going to run from 5' over here to 3' over here. And it's going to be synthesized in this direction. And this is referred to as your lagging strand. So I'm going to first talk about your leading strand because it's a little bit easier to see. So here your helicase is going to come in and open up your parental DNA. This is going to be your template DNA, and this is going to be your baby blue is going to be your complementary DNA. Or complementary strand. So you're going to work from your DNA polymerase 3. So it's shown here binding here, but I'm just going to pretend that it's shown binding here just so I have more space to work with. So your DNA polymerase is going to come up here and bind to your leading strand, and then it's going to start synthesizing the new complementary strand, so putting those complementary base pairs, so guanine, cytosine, thymine, all of those in this direction as shown here in this black arrow. So it's going to come in this direction, and it's going to be continuous. This lagging strand's not so straightforward. You guys will probably discuss this a lot more in lecture, so I'm not going to go too into it here. But essentially, helicase is going to open this up. Your DNA ligase is going to, or not your DNA ligase, I'm sorry. Your helicase is going to open this up, and then you're going to have a, I'm going to start off here because I think this is a good spot to show it. Your primase is going to come in and put down a primer. This is made out of RNA, not DNA. So this little primer is going to be here. And then your DNA polymerase 3, which is this peach enzyme, is going to recognize this primer. And it's going to essentially build in this direction, the direction of this arrow. So from 5' to 3'. Because remember, this is running from 5' to 3' this way. So it's going to build little fragments in this direction. So your DNA polymerase recognizes this primer, comes here, and starts building the complementary bases here. And then it kind of falls off, and someone else will eventually come in and recognize, so it's actually your DNA polymerase 1 will come back, recognize this little RNA primer, and take these RNAs out and put DNAs there. And then it basically continues at every area that there's a primer at. And eventually you'll have a continuous strand. So that's actually really hard to explain without a video. So I did put up a video for you guys to watch. Let me see if I don't think I'll have to tag it in the other, I'll put it up for you guys, but there's a video that I want you guys to watch that basically shows this happening simultaneously, and it's a lot easier to see than me trying to verbalize it. So now it's going to be your turn. So you have, you're going to replicate a strand of DNA in the next slide. So this is just labeling everything. So these purple P's are going to be your phosphate group. This pentose structure is going to be your sugar. This one's going to be adenine, thymine, cytosine, guanine. Remember, adenine and thymine interact with each other. And how many hydrogen bonds are formed between those two? 2. And then cytosine and guanine interact with each other. And how many hydrogen bonds are formed between them to stabilize those structures? 3. Okay. So here you basically will, see if this, so you're going to drag these over here. You're always going to go in the 5' direction to the 3' direction. And you just have to drag whatever is complementary. So always notice that the phosphate groups are going in opposite orientations. These pentose sugars are pointed in opposite orientations also. Remember, they are anti-parallel. So you'll go ahead and practice that. And that is essentially activity 1. So activity 2, you're going to be demonstrating mitosis with, if you have like plastic forks and knives at home, you can do it with that. But if you just want to like draw it out, draw each stage out of mitosis, that works also. So we didn't quite discuss mitosis yet. So if we go back, let's see if I can go back without making this so difficult. We go back, we talked about interphase. We talked about what happens in the G1 phase. Everything that I talked about in terms of DNA replication happens in this S phase. We talked about what happens in the G phase. We talked about, and that is shown here too. We talked about different checkpoints. We talked about what is basically telling the cell cycle to go and initiate, what is suppressing it. So this was all about the S phase. But the thing we haven't talked about is what happens in this M phase, And in your lab notebook, it does show these M phases. It shows these M phases as prophase, metaphase, anaphase, telophase, and cytokinesis. So I refer to these as PMAT, is how I memorize them in terms of the order. Okay, so now we're going to go over mitosis. So mitosis is a nuclear division that conserves the chromosome number by equally distributing the replicated chromosome to each of the daughter cells. Remember, everything is identical here. What is shown in this cell is identical to what is shown here and here. So the significance or the purpose of mitosis is asexual reproduction, growth and development, as well as cell replacement. And cell division occurs on a continuum. It's a dynamic, it's very dynamic, and it exists on a continuum. You start off with interphase, which is shown here. Interphase, like I said, is where your cell is going to grow. It's going to make all the necessary organelles to be able to have support two cells in the end. So you're going to also have the DNA replicated. And your DNA is shown here as this like spaghetti-like loosely formed DNA that is encased in this nuclear envelope. As we progress, there's going to be changes that occur to these cells. And these are the, shown here in this navy blue, are the M phases. We have prophase, metaphase, anaphase, telophase, and then cytokinesis. So PMAT. So we're going to go through, I'm going to let you guys read this one on your own. I'm going to go through them one by one in the next slides. So here we are showing prophase first. Okay, let me pull up. So in prophase, your DNA is loose. It's like spaghetti-like is what it's usually referred to as. You have two chromosomes, and here it's shown as blue and purple. And your DNA is enclosed in the nuclear envelope, and you have your centrioles shown here. The first thing that's going to happen is that your chromosomes are going to condense into the sister chromatids. So they're going to get nice and tightly packed in these sister chromatids, which are shown here. And this is going to help because in order to separate properly, you need to condense that DNA. You're also going to have the nuclear envelope get dissolved, broken down, degraded. There's going to be certain proteins that initiate this response, the disassembly of the nuclear envelope. So as you can imagine, if your DNA is enclosed in here, it's really hard to separate it. We need to get rid of this, this structure in order to separate our DNA. And these centrioles, you're going to see them to start having these microtubule organization centers, or MTOC. You're going to see these spindles forming. So as you can see, they're going to be like these microtubule organization centers or spindles are going to be forming. And as these form, this kind of happens, this is the transition to the metaphase. So as they form, you're going to see that the polar portion of the microtubule is going to connect to the chromosome. It's going to specifically connect to the kinetochrome, which is this protein structure that is found right next to the centromeres. And once they connect, these spindles connect there, are these polar microtubules. So this shown here is the polar part of the microtubule, and these back here are the atrial parts of the microtubule. So once they connect, you're going to see them line up here in the midline. So you can think of prophase as P, preparing, prepping. So remember, it's PMAT. So the prophase is preparing, it's prepping, it's getting ready for cellular division. The metaphase, the metaphase, think of it as the midline or the middle. So all these sister chromatids are going to line up in the middle. The microtubule is going to connect to them here on next to the centromere on the kinetochromes. And at this point, all your chromosomes have become lined up at the equator of the fully formed microtubular spindle. At this stage of the mitosis and the cell cycle, chromosomes are in their most tight condensed form. This is also sometimes referred to as this midline is also referred to as the metaphase plate. So now we are getting ready to really separate these things. So now we move on to the anaphase. So here, think of anaphase, think away. The centromeres are going to separate and the sister chromatids begin to move to the opposite ends. So this half is going to come on this side and that half is going to go on that side. So this here the centromeres split, the sister chromatids begin to separate, and you essentially have these motor proteins that they're called dynein and kinesin. They basically walk across these tubules and they move the chromatids towards this part of the the centrioles. They're just pulling these away, and then you go on to the telophase. So here in the telophase, there are two clusters of chromosomes which decondense. So the condensed DNA is going to begin to decondense. The nuclear envelope is going to begin to form again. And you will see that this little pinching occurs here where it begins to, I guess, bud off. And this is referred to as a cleavage furrow. And you're getting ready to have two cells, essentially. So your nuclear envelope's back, your nuclear DNA is deconsensing, you're getting this cleavage furrow, pinching, and essentially in the end, you have cytokinesis occurs, and that's cytokinesis is when you have this pinching occur, and then you have two identical daughter cells. Each is a diploid. Its nucleus has two of each type of chromosome, just like the parent cell. And now it could go on to interphase again, if it wanted to. It could go on to interphase, or it could go to the pit stop spot that we talked about, that quiescent G0 phase. Okay, so those are essentially the different phases of mitosis, and here's a summary for you guys to kind of go over it and start thinking about these. So remember PMAT, prophase is a prep phase, preparation, metaphase is at midline, anaphase is away, telophase is when you have your chromosomes decondense and your nuclear membrane forms, cytokinesis is when you have that cleavage furrow occur. So the process of cytokinesis is splitting of the cytoplasm, and this actually differs between plants and animal cells. So like I said, you have this cleavage furrow, and this occurs in animal cells, and you essentially have this cleavage furrow that you have actin and myosin that are initiated here to allow this action to occur, where you will end up with two identical daughter cells and you have the splitting of the cytoplasm. In plant cells, however, this is a little bit differently. So in plant cells, you don't have centrioles, and cytokinesis occurs differently in the plant cell. During cytokinesis, in the plant cell, you can't have that furrow or that pinch off because of the cell wall. Remember, we have our cell membranes, plasma membranes, things like that in animal cells. In plant cells, we have cell walls that are a lot thicker. And because of that cell wall, instead what happens is a cell plate will appear in the center, which is shown here. A cell plate will appear in the center of the spindle fibers that is formed from vesicles produced by the Golgi body. These vesicles release cell wall material that will be linked together to make a new cell wall. So this is a time-lapse video of a cell dividing, and this takes 90 minutes. So this would take place in 90 minutes. It's just been sped up, and that is just a time-lapse video of a cell dividing. So activity 3, you're gonna identify the different phases of mitosis. So the different phases that occur during the M phase of the cell cycle. So ideally, we'd be in lab using microscopes to look at the cells that are undergoing mitosis, but instead we have taken pictures from slides. We have pictures of the root tip of onions, and we also have pictures of the blastula of whitefish, and they're going to be undergoing mitosis. And essentially what you want to do here is you want to look at these images and try to identify all the stages of mitosis. So you want to well, actually the cell cycle. You want to identify the prophase, you want to identify the metaphase, the anaphase, the telophase, but you also want to identify the interphase, or which image best represent those phases. So I found these images online that identify this in an that onion cell that you guys are also going to be looking at. So as you can see here, this is the prophase, this is the metaphase, it's all lined up. This is the anaphase, they're pulling away. And this is the interphase where the whole growth and DNA replication occurred. And then they're identified over here for you as well. The only one that's not identified on this side is the telophase. So here is the telophase. You see that cell plate beginning to appear here. So go through these images here and try to look like at different parts of this and identify like what stage is this? What stage is that? And try to find an image that represents all the stages we talked about: interphase, prophase, metaphase, anaphase, and telophase. So here's some more slides that you can choose from. Here's this side, and this slide. These are all those onion root tip slides. So there's 3 slides you can look through. And then you're also going to do essentially the same thing, but you're going to do it in animal cells. So here you're going to be looking at whitefish blastula. And you're going to, once again, identify the stages of mitosis. In your lab manual, there's a page where you will draw them to the best of your ability. You will do this for the plant cells as well as the animal cells. So I found this image on the internet, and I labeled these two according to what I believe is happening. So I labeled this one as interphase. So here you have that nuclear envelope showing. Your chromosomes are kind of like spaghetti-like. They're not quite dense yet. And then I labeled this one as prophase as well as this one. So here you see the chromosomes appear more condensed. You see that they're getting a lot darker. They're condensing. And then you also see that the nuclear envelope is beginning to disassemble. It's not apparent anymore. And then after prophase is metaphase. So remember, they're all your sister chromatids are going to line up. The spindles are going to attach. And I identified this one as metaphase. And then anaphase, these sister chromatids are going to start getting pulled away from each other onto the opposite ends. They're separated and moved towards the poles, so these centrioles. And I identified this here as anaphase as well as this here. And then there's telophase. So I believe this here is a good depiction of early telophase at least. So the chromosomes are at the poles and are becoming more diffuse. Here you'll begin to see the nuclear envelope begin to reform, and you should also begin to see the cytoplasm close in, and the cleavage furrow happening, occurring. This is more of an early telophase. I do think there's some images in here that are late telophase. So something like probably here is a good late telophase image. But go through these and see, like, what would you consider this to be? What would you consider this to be? This one, something like here, what would this be? So just go through all of these and try to identify them. And that is it for mitosis. We will cover meiosis next, and I'll try to put that up on Thursday.